Hi, this is Miss Arnold, and you're going to be taking notes on how Hina end up with too many blast cells. So you'll be using the handout to take your notes. That's titled Introduction to Cell Division, where we'll talk about cell growth and how cells divide. So for the first part, let's learn some facts about cells. So this is part of the cell theory. And the cell theory has three parts. The first part is that all living things are made of cells. Cells are the basic unit of structure and function of living things. So this is for single-celled organisms all the way to multicellular organisms that have different tissues and organs. It all starts at the cellular level. And lastly, all cells come from existing cells, which is kind of what we're going to focus on in these notes. And at this point, you should pause and watch the YouTube video to give you a little bit more history about how the cell theory became about and the sort of the research and the scientists that are involved in creating those statements. Okay, so going back to our question, well, if all cells come from existing cells, then Hina's extra blast cells must have come from other cells. So that's kind of what we're getting at. So why would your body need new cells in the first place? Well, your body is growing. I'm going to delete that there. Your body needs to replace old cells, and your body also needs a way to repair damaged cells if you get an injury or a cut or something. So you need new cells for all of those purposes. So how does your body make more cells? Well, as cells grow and divide, they go through a process called the cell cycle. Now there's two videos that you're going to watch that show cells going through the cell cycle. And on your sheet, you're going to record your observations and your questions. So at this time, go ahead and if you're in the slides, go ahead and click on the video links to watch the videos. And as you're watching it, so there's no sound, but what you're watching is the cells of fruit fly embryos dividing. And they have to grow really quickly, so their cells divide very quickly. And the reason we're able to see these is because the DNA inside the cell is tagged with a fluorescent dye that glows. So in this video, they might appear white. That's the actual DNA that you're seeing. So as you're watching, make some observations about what you see happening. The DNA in the cells that have been tagged, so what's going on with the DNA? What do you notice? Okay, in the second video, we're looking at mitosis in pig, I believe it's liver cells, kidney cells. And again, these have been tagged with a fluorescent dye and used computer software to sort of enhance and make them appear different colors so you can see what's happening inside the cell as it's dividing. So again, write down your observations and any questions you have. And specifically, the DNA is shown in red. Okay, so let's consider the life of a skin cell. So, your skin cells are ones that divide pretty quickly because your body is constantly needing to make new skin cells for the ones that come off, like when you wash or, just carry out your day-to-day activities. So during its life, a skin cell will grow, perform functions like that provide structure and cohesion that allows our skin to protect us, and it receives the signal, it'll divide in order to replace old or damaged cells. So cells don't constantly divide like the cells that you saw in the previous videos. So that process of division was sped up for the video. Especially in adults, certain cells rarely divide. For example, your brain cells, pretty much once you're an adult, they are no longer really dividing. And cells that divide go through a specific set of steps known as the cell cycle. And the speed of that cycle, again, could be different for different types of cells. So this is the general cell cycle. So these are the steps that each cell will go through. And again, it's different for the different types of cells, but they generally go through all of these phases. So there's two phases that we'll be looking at, mitosis and interphase. And you can see mitosis is relatively short to interphase, which is much longer. And during interphase, the cell size grows in size and duplicates all of its different parts, including its DNA. And I'm going to fix the coloration because I just realized it's really hard to see the remainder of this ring. The interphase ring is the same color as the background. But yeah, that should be about, I would say, 3/4 to maybe like 80% of the cell cycle is in interphase, and then the rest is mitosis. So during mitosis, that's where the cell is actually going through the complex task of dividing all of its parts. And mitosis itself is divided up into smaller phases that we'll talk about later. But you can see, like, at the beginning of mitosis, you have one cell, and at the end, you end up with two identical cells. All right, so here we go. So this is actually, you can see the full cycle. So interphase is divided into three parts. You have G1, which is the growth phase 1, where cells grow. And then during the S phase, that's where DNA is duplicated. And then there's another growth phase 2, where the cell gets ready for mitosis. So it just makes sure that it has enough of the organelles that are going to need to be duplicated, and then it'll go through mitosis. And then it starts again. Cells can also exit the cell cycle and go into what's called a resting state, or G0. Scientists are still learning about G0 and how cells decide when to enter and exit this resting state. So, they might not be growing or duplicating, but they're just sort of like resting and doing their normal functions. And this might be, some cells that don't divide very often might stay longer in this state. Okay. And then, like I said, we have mitosis, which is the step where the single cell divides into two identical daughter cells, and each cell has a copy of the same DNA that the original cell had. So these produce body cells, but not sperm and egg cells, which are created using a different process that we'll learn about later in the year. So an important note is that different cells divide at different rates, which we kind of mentioned before. So that rate really depends on your body's needs for that cell type, the job of that cell, and how long its lifespan is. So for example, your skin cells have an approximate lifespan of 2 weeks, and then they need to be replaced by new cells. So those, again, are dividing pretty frequently in order to create those new skin cells. Your red blood cells, on the other hand, their lifespan is more around 4 months. So again, those are being made in your bone marrow, and your bone marrow is replenishing red blood cells at that rate. Your liver cells are good for like maybe a year and a half, so 300 to 500 days. So not as frequently. Your intestinal lining, however, you can see that this is the part that lines and comes into contact with all of those like stomach acids and enzymes that's helping digest your food. So those have a very short lifespan of only 2 to 5 days, just because of that kind of harsh environment that they're exposed to. So your body is constantly needing to replenish those and is shedding the dead cells through your waste. You know so that's a very fast reproducing and dividing cell. Whereas other parts of your intestine, like the muscles and other tissue, could be 16 years before they need to get replaced. So, and some of the cells that divide, the most rare or even sometimes never, are your brain and spinal cord cells, or your neurons. So those typically remain in that resting state and rarely divide, especially once you reach adulthood, which is oftentimes why they say, like, if you lose your brain cells, you kind of lose them for good, because you don't typically get to replace those. So it's really important to take good care of your brain cells. And for a cell to divide into two healthy, identical cells, the final two cells needs all of the cell parts necessary to function. So just think, like, what cell parts would a cell need to duplicate before it can divide? So think about the important parts of a cell that we learned about before, right? Thinking about, like, what's in the nucleus. Think about energy, right? So all of those things. And of course, it's going to need the instructions. So each new cell is going to need to have a copy of all of the instructions that are needed to make other new cells in the future. So this is, of course, is DNA. So DNA contains all of the instructions for a living organism to function. And if a mistake happens during DNA replication and it's not corrected, then your cells do have a way to, cause that cell to sort of self-destruct, or we'll call it cell death. Or if it's not corrected, it could lead to cancer if those mistakes happen. So during interphase, the DNA is contained within the nucleus of the cell, where that's when it gets duplicated during that S phase to prepare for mitosis. So if you can imagine a ball of yarn that contains 23 pairs of strands, each pair a slightly different length, without a defined procedure, it would be really difficult to find the pairs, match them up, make sure that both cells, the original one and the new ones, got a complete accurate copy of all of the pairs. So they're kind of all sort of jumbled up in there like a big, like I said, ball of yarn, or like spaghetti. And then your cell needs a way to kind of organize those in order to get ready to make a copy. So part of the way the cell does this is that during mitosis, the cell organizes DNA into bundles called chromosomes. This organization allows the cells to copy and divide its DNA without mistakes. I like to think of it like if you're getting ready to move, you first want to pack your things into boxes and kind of organize them by like the room that they're going to go into, right? You don't want to just like have all your stuff and bring it over bit by bit. You might forget things can get lost, misplaced. So this is kind of like what the chromosomes do. It helps organize the DNA to get them ready for copying and division. Okay, so that's where we're going to end for today, and then next time we'll look more closely at those steps within mitosis that lead to